Welcome to ASM Live at the 115th American Society for Microbiology General Meeting in New Orleans, Louisiana. We're here today to talk about a number of stories related to microbiology that are important, interesting, and sometimes downright surprising. Today, we're going to be talking with our guest, Stephanie Momeni, and she's going to tell us about some work that they've been doing with a bacterium that lives in our mouth, Streptococcus mutans. But before we start, I'd like to remind the audience to please ask questions. Go up to the microphone, introduce yourself. We'd love to hear your questions and join in on the conversation. If you're listening online, please send any questions via Twitter at #ASMLive. So Stephanie, tell us a little bit about the research you're doing with strep mutans.
Actually, I'm part of a much larger group at the University of Alabama at Birmingham in the laboratory of Noel Childers. And we have been doing an epidemiological study of Streptococcus mutans related to dental caries in children and their household family members over the last 8 years. And during the course of that, we're using a genotyping method, RET-PCR. And my dissertation work was focused on multi-locus sequence typing. And as an aside, we got today's data that we're going to talk about.
So you talked about a couple of things just to make sure everybody knows what they are. Mm-hmm. Why do you use the PCR approach and the multi-locus sequence typing?
Okay, so the RET-PCR approach looks at different parts of the genome. It looks at the intragenic regions that aren't really expressed or proteins aren't generated from those. It creates fragments, nice banding patterns. Multi-locus sequence typing looks at the housekeeping genes, which are highly conserved that don't change very much over time. So it's just two different parts of the genome that we're looking at.
And what are you trying to find from these approaches?
Well, what we would like to be able to find is certain genotypes associated with dental caries. That was our primary focus. Of course, now we have perhaps a new focus, other health issues that may be associated with strep mutans, but primarily looking to see if we see certain strains associated with that, dental caries.
What's the difference between the different strains?
Between the different serotypes?
Yes.
Okay. Between the different serotypes, it's the cell surface proteins that are on them are slightly different, and that changes how they interact with the human immune system. So.
And how many serotypes are normally seen?
For strep mutans, there are 4 clinically relevant strains. The most recent strain, there's C, E, and F. The most recent strain is K, which was recently discovered in 2004 by a Japanese group. So.
And your focus is on K.
Well, actually, like I said, my primary focus is actually multi-locus sequence typing. And during that, when we went to publish, I thought, let's just go ahead and serotype these strains and see what we have. And essentially what we found was a little surprising. We found quite a bit of serotype K, which to our knowledge has not been reported in the US, at least not that I was able to find, at least not in publication. And so we were really interested in that, and that started us digging.
So why is serotype K so important?
Well, it had, like I said, it was only recently discovered in 2004. So there are new studies coming out of the Japan group looking at it. To my knowledge, it's only been isolated in Japan, Finland. The UK has some, and a recent paper just came out for Colombia. And essentially, it's important because they're finding a relationship between it and systemic diseases such as endocarditis and hemorrhagic stroke. Those are just a few.
Is there something particular about serotype K that might be associated with these diseases? They're
learning a little more about it. Like I said, they have very few isolates to work with, and but they are conducting some studies. And what they have found, most recently, a paper came out earlier this year where they found that the protein antigen, it's the surface protein that allows it to adhere to teeth, for instance, in the oral cavity, is defective in serotypes F and K. I don't want to not talk about F because F is also important. We simply didn't find it in our study, but that's why I focus on K more. But F and K both seem to have this defect. And if that's true, it helps it evade phagocytosis in the immune system. and also helps them to invade human endothelial cells.
But if they don't stick to the teeth,
Right.
they're still associated with caries, though.
Well, and they can have carriogenic properties. What makes them special is a collagen-binding protein. Actually, there are two different ones that they've been identified so far, and those are CNM and CBM, and those allow them to invade the human endothelial cells. So not all serotype Ks can do that, but those with the collagen-binding proteins can do that.
Do you have an idea about the frequency at which somebody who carries one of these serotypes with the collagen-binding proteins, how often those people come down with one of these systemic diseases?
And like I said, that's still very, very new. We do know that, streptococci are very commonly associated with endocarditis. That's been well documented since the 70s, and bacteremias as well. And now that we're looking, this new serotype has come into play, they need to go back and look at that data to see which, which serotypes were actually causing. And the prevalence of endocarditis, for instance, is maybe 3 to 9, depends on which paper you read, percent, at 3 to 9 individuals out of 100,000 individuals. So it's relatively low. And then a certain percentage of that, maybe 15 to 20%, are actually because of strep mutans. So it's a relatively low number. But now that they have the collagen-binding proteins, they need to look at that and start looking more closely at people who have endocarditis and see how many of them. But collagen-binding protein, CBM, is most associated with serotype K and endocarditis.
Have these serotypes changed over time, do you think, or are people just discovering the new serotypes?
I think it's just recently been discovered is the issue. And there may even be a new serotype, a paper I just found this morning, and then also in our study, we had 2 isolates we couldn't identify with the PCR detection method for the serotypes. And so I found another paper this morning that also had that. So there may actually be another one, but it would be very minor. It doesn't occur very often, at least to our knowledge.
Do you have an idea if the systemic disease might be decreased by simple oral hygiene types of approaches, like brushing your teeth regularly?
Well, actually, that's a good question. Honestly, because the serotype Ks and F are rare, they're only like 5% found in the oral cavity, obviously because they can't adhere to the teeth. But, there's no guarantee that that would necessarily help, but good oral hygiene is always a good thing, and it does help improve, your risk, especially flossing, to help break up the plaque.
I think that although this has been known for a long time that oral microbes can have systemic impacts, it's not something that most people think about.
Right, and oral health is really, really important, and they're finding, especially with the microbiome studies, they're finding a much higher correlation between, your oral microbiome and what happens with the rest of your body. The one that just stands out in my mind is nucleatum. Sticks to the teeth. It's about 4, I want to say, if you go 4 days without brushing, it tends to show up in a high number, and that can affect your children and birth rates and problems with that. So it can have an overall health effect.
That's an interesting thing when you think about human evolution and you go back to the time before people brushed their teeth.
Yes. And there is a disparity between industrialized nations and non-industrialized nations. So in industrialized nations, we have a lot more sugar in our diets, and that aids, strep mutans love sucrose, and that's how it sticks to your teeth, is it synthesizes the sucrose, and it creates a polymer that allows it to stick to teeth. So we have much more of a problem than a country that doesn't have easy access to high fructose corn syrup, for instance.
I'd like to remind the audience, please ask questions if you would like to. You can just step up to the microphone. And anyone listening online, you can send a question in via Twitter at #ASMLive. One of the interesting issues is these collagen-binding proteins that you mentioned have this real strong correlation with the systemic disease.
Yes.
In the heart or the hemorrhage in the brain. And do you think that there is a rationale for why a collagen-binding protein would have that association other than just the dissemination?
Well, I think, at least in my most recent reading, is it has an effect on platelet binding capabilities, so it'll cause you to bleed out more easily, which is how the hemorrhagic stroke works, I guess you could say. So yeah, I think that's because those proteins have an effect. It's been known that the serotypes do have an immunological effect on the body, immune defense, basically heightened immune reactions. So basically with the serotype Ks, they also can evade the immune system. So, you know, not only, they can make you stay sick longer, basically.
The serotype K that does not carry these collagen-binding proteins, does it have any association with systemic disease?
Actually, for serotype K, not all serotype K carry the collagen-binding proteins, but the ones of most concern are those that do, in particular CBM. So CNM is fairly commonly found, about 20% of strep mutans isolates, whereas CBM is less frequent. But whenever it is found, it's more highly associated with disease.
So I never let you finish telling about the statistics that you found in the school children.
Yes. So for our group,
Yes.
Essentially we did just randomly selected 100 isolates, and we didn't limit it to one per child. We wanted to see stability as well. And what we found is that we had 57 children at the end of the study that, you know, had individual isolates tested. And of those, we had 10 that had serotype K, which gave us a prevalence of 17.5%, which is the highest noted except for an Indian study. And the difference between their study and our study is we used individual isolates, bacterial isolates, as opposed to whole saliva, which will give you the whole profile for the child. So ours is much a truncated view, if you will, as opposed to the saliva view, which gives you the whole profile.
If you go back and you look over time, does the profile tend to stay the same? If you have serotype K, do you continue to maintain serotype K?
And that's what we want to look at next. Because like I said, this was a last minute thing we did on our paper. And now we're going back and we're going to screen all the saliva profiles for the children in both of our studies that we have, and look at an earlier period and a later period to see if the profiles do change over time, but also if they acquire the collagen-binding proteins over time. Because as we said in this initial study, we did not find any that had collagen-binding proteins.
We have a question from the audience.
Sure.
I think Gary Terrances from the University of Puerto Rico. I was going to ask about how you picked your subjects, because there's also I would imagine there seems to be a strong correlation between ethnic groups and microbiota that you may have in your mouth. If I remember, for example, I lived in Africa for a long time, and from a very young age, people break down certain types of branches, and they spend the whole day brushing their teeth. And I would imagine that under certain circumstances, those that cannot bind to the teeth can always get trapped in the interdental areas as well, so they would not really need any type of an adhesive, so to speak.
Right. So for these, especially although infection through dental, going to see the dentist or flossing, for instance, which make your teeth bleed, is relatively low, or that they haven't found any strong relationship there. It is possible that if you're making yourself bleed during this and you've got serotype K in your plaque, for instance, you could be introducing it into your bloodstream that way, and that is one of the theories of how it gets into the bloodstream. To answer your original question, which was about the study population, essentially we have a very small group in a small rural town in Alabama. It's all for this particular portion, it's all African American children. So it is very isolated. It may not be generalizable to the general public, so it shouldn't be assumed that all African American children will have the same results that we have. But it is a very isolated group, and made us start thinking, well, hopefully somebody else out there will hear this broadcast who's working with strep mutans, and we would encourage them to please serotype them so we can find out, if there's a lot of serotype K in other populations in the U.S.
Did you guys say what age group you picked from, and have you does age really matter when you're looking at
And that's one of the things that we want to look at is whether, when they acquire them and over time to find out if it does matter. So that's an excellent question. The age groups, we actually have two different cohorts that we work with. The first one, we started with school-age children 5 to 6 years old, and we have followed them every 6 months and then annually for the last 8 years. The second cohort were infants, and we followed them from time of colonization to last 5 years, I believe. We started that cohort later, so, okay,
Thanks.
thank you.
Is the serotyping approach easy enough that this could be done more broadly?
Oh yes, there is a PCR approach that has been developed, but as I said, we have found a few strains that aren't typable by this, and we don't know if that's because it's a new serotype or if it's sensitivity of the method. But there is a published protocol for C, E, and F, and then in 2004 they developed one for K as well. And our approach, we used a cyber green approach, which was really nice because you can run it in under a couple of hours and essentially get the profile, but we worked with individual bacterial isolates. It works well for that. We're working on a multiplex that yields a really nice result, and those do have to be run on gels, unfortunately. So it's a little bit longer to do that.
If I recall, the entire genome of several of these serotypes have been determined.
Yes.
How closely related are they, other than the genes that you've mentioned so far?
Well, strep mutans, and that's a kind of an interesting question. They are highly diverse, but they're very similar.
That's a straightforward answer.
It sounds like an oxymoron, but there's, relatively, it's a monoclonal population, that, speaking genetically, but they essentially are very similar, but there is quite a bit of variability within the genomes. So, especially within for the serotypes, what differentiates those is the glucose polymer, rhamnose glucose polymer interactions on the cell surface, and the length of the glucose, how many glucose side chains it has.
But within, for example, serotype K, are they more similar than they would be between K and A or E or what have you?
Yes, yes. They're more similar within K. And for instance, in our study, like I said, we used the cyber green approach. We found that C was pretty stable in the peaks that we got for it. E and F, very stable. K, we get a little bit of shifting in that. So we thought that was very interesting in their melt peaks. So there is some variability within K.
So I asked that question because you might think that in the oral microbiome, there could be a lot of opportunities for gene exchange.
Oh, yeah.
Where you could share some of these traits between the different serotypes.
Right, and that's one of the theories of why we think they may acquire the collagen-binding proteins over time, is strep mutans are very naturally competent, so they like to take up other DNA and incorporate it into their genomes and make it useful.
Do you see any practical applications of knowing this information? What would you do differently now that you know this versus before?
Well, for right now, it's still very early days, but, it may be possible that we could screen these children and develop a screening method where you go into your dentist and we take an oral sample. We are able to run a PCR, in a couple of hours and let you know if you have this serotype K. If we do continue to see more and more relationships with serotype F and K related to systemic disease, that could be very helpful in letting people know, you have a higher risk of endocarditis or bacteremias, so you want to be careful when you floss.
Do you have an idea? Do you have a guess why different serotypes would be more prevalent in different groups of people? So there was a question earlier about the ethnic association, and you could imagine that certain serotypes might be prevalent in people with different immune properties.
Right, so humans are so variable, so I definitely think there could be associations related to different ethnic groups. For instance, when we started talking in our group about doing the whole saliva sample, there was a lot of misconception that most people only have one serotype. And I went through the literature and found that's not necessarily true. There are some people who have more and some that have less, that only have one. And actually, I was just reading on my way over here, there was an interesting paper, and they were saying that the cardiovascular risk was much greater for people who had multiple serotypes as opposed to one serotype.
That's interesting.
So.
You could imagine that a reason for that could be that it could help stabilize serotype K in the oral cavity, and so you would have less of a transient exposure in that case.
And actually, one of the things we're theorizing with our larger study of strep mutans in general, where we're looking at the epidemiology related to caries, is we find that there's a stronger relationship if you have more different genotypes than if you have a single genotype. Your caries incidence is much higher, for instance. So we think that there may be some competitive relationship going on there that may lead to a higher immune response from the host.
That makes some obvious implications about the idea of using probiotic types of approaches and to reduce certain types of serobars.
Yeah. So, yeah, probiotics, I'm not sure if that would really work for necessarily the oral cavity. It's great for the gut microbiome, but I'm not so sure about if it would get rid of your strep mutans.
Why not? You could have a gargle that you use with
Strep mutans are pretty hardy. For instance, they're very sensitive to antibiotics. So when you take antibiotics, you probably kill off most of them, but they tend to come right back. So it's amazing how well they stick around.
How did people happen to discover in Japan this association with serotype K?
That's an excellent question. They actually had found 2 strains that they could not identify through a traditional immunodiffusion method. And essentially what they found was, one was from a Endocarditis patient, the other was from a patient that had bacteremia and also a subarachnic hemorrhage. And they had isolated these 2 isolates and they couldn't identify them. So they went through a number of tests to prove that it really was strep mutans. But the key thing that stood out was it actually lacked the glucose side chains, which is what has been the identifier for CE and F. So it either has none or very little of the side chain. So.
And it was first discovered specifically in the case where people had more serious systemic disease, and then they went back and looked more broadly?
Yes, yes, and they have done a lot of work with serotype K, but even still, it's fairly rarely found, so we have a very limited number of isolates to work with. So, if other people are working out there with strep mutans, we would encourage you to serotype your samples, because if we can find more serotype K from other populations, that could be very helpful.
We have another question from the audience.
Hi.
This is Rahman Hariri from University of Pittsburgh Medical Center. Have you looked at to see the population change of strep mutants in children and adults and elderly? Because we see more endocarditis related to strep mutants in the elderly populations.
Yeah. In our population, we're mainly looking at the child and the household family members. We do have some grandparents mixed in with that, but we haven't made it that far in our analysis to really look at, right now we're just looking at mother-child and primary caregiver, but we're hoping to extend that to the household family members and might be able to answer a better question. The other thing is when we go back and serotype the whole saliva samples, when we find children that have this high prevalence of F or K, we want to go back and look at their family members and see if they have them as well. So, thank you.
I think that's an interesting question. Endocarditis may be a little different, but it's also possible in some of these cases that an infection earlier in life can result in a change in the host that has long-term complications as well.
Right, and as I said, these children that we've tested did not have them, or at least the few that we did. And you have to realize we've got a catalog of 30,000 isolates we could look at.
You better get to work.
Yeah, and roughly somewhere in the neighborhood of about 200 children that we're going to screen with the saliva study. So that's maybe next year's meeting. But it's a very interesting point that you made. I have tested some of the adult samples. Basically everything I've run MLST on now, I've gone back and serotyped. And not part of today's poster, but we did find that it's more common in the adults that we start to see the collagen binding proteins. But it's a very small sample size. So.
Well, it's been really nice talking with you. I'd like to thank the audience for joining us today and for your questions. Everyone watching online, we appreciate you joining us. Hope you come back again for another episode of ASM Live. Here, the 2015 American Society for Microbiology meeting in New Orleans, Louisiana. Thanks very much, folks.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shower.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.